Now I'm going to talk a little bit about the difference between LSM780 and LSM880. So first, both of them have confocal modules, but our LSM880 is also equipped with super-resolution modules, which are SIM, PALM, and DStorm. Basically, super-resolution means higher resolution than a confocal microscope. So if you are interested more in intracellular level, for example, organelle structures, sometimes confocal might not be enough. Then in that case, you can think about using super-resolution technique. Please consult with us in case you do have experiments that you would like to get higher resolution than confocal. They have both their pros and cons, so we're going to talk about it a bit more if you really need the technique for your experiment. Okay, so the first difference between LSM780 and LSM880 is on the locate. So you're going to see that here we only have 3 buttons. So we have multicolor, bright field, and closed shutters. For any fluorescence, you're going to click on multicolor. Multicolor can only allow you to see green fluorescence, which is VC or your GFP, or red, which is your rhodamine or your text red, your RPE. So you will need to use those 2 colors to find the area of interest or find the focus. If you need to use DAPI to find the area, then you would want to use LSM780 because LSM880 doesn't have a filter for the eyepiece for you to see DAPI. You can always image DAPI channel with lasers, so all the lasers are exactly the same, but just in the eyepiece is a bit different. Okay, so the multicolor allow you to see green and red fluorescence simultaneously. Sometimes it will be really useful, sometimes it might not be, especially if you are trying to differentiate which cell has green and which cell has red. And one fluorescence is very, very bright compared to the other channel, then you might not want to use this because the very strong fluorescent channel might mask your weaker one. If you're trying to do colocalization, sometimes it's very useful to use this system because the multicolor allow you to see both green and red. So if you do want to find the cell where 2 proteins are colocalized, you can see the 2 colors at the same time. So it's relatively easy to find which area you want to image. Bright field is your bright field image, not image, your bright field eyepiece in eyepiece. So keep in mind that 880 doesn't allow you to take bright field pictures. You can see bright fields using this, but it doesn't allow you to take bright field pictures. Okay, so if you do need bright field pictures, then you want to use the 2 LSM780 that we have. Otherwise, to adjust the brightness is exactly the same. So you click on the light source, okay, and you can increase or decrease the light intensity. Once you find the area and find the focus, then you can click on closed shutters to shut down everything, and then you go to acquisitions. Okay, in terms of acquisitions, they are very similar. The speed for 880 is a bit faster than 780, but probably not a very big observable difference. Okay, otherwise this interface is exactly the same as our LSM780, but keep in mind we cannot image a bright field picture on 880. The other thing is for the argon gas, you can see that here we do have the power on, off, and standby. This is because on this system you won't find that little switch that go from idle to run or from run to idle on 880. It is controlled through the software. So once I click on the laser, the 488 nanometer laser that's for our GFP or our VC will be on. If you're the first one using the system, This one would require warm up. Once you click on, you'll see like a red, like a red color on 48. Then it means it's still trying to warm up. You cannot use this laser yet. Okay. Once the red color disappears from here, then you can use the 458, 488, and 514 laser lines. The third difference is the chamber itself. So if I move aside, you can see that this chamber is black. Okay, so this is a chamber that would protect your sample from the light. So technically, you can image during with the light on. However, if you look through eyepiece, it's going to be a bit harder to see your sample. So we generally recommend that you turn off the light. If you want, you can keep the light on. It's totally up to you. Okay. The other thing is those doors are actually light sensitive. This one, and this one. Okay. So make sure that they are closed completely. If sometimes you think you actually close it, but it's actually not closed, okay, the laser would not start. There is a laser protective feature in this case. So if you find that when you click on live, there's no signal at all, you try to increase the gain master, it doesn't work, then it does often mean that the doors are not closed properly. So make sure that this one, and this door here are closed properly. Otherwise, you wouldn't be able to start imaging. Thank you.